Scripps Institution of Oceanography at the University of California, San Diego. Meet Scripps Director Charles Kinnell.
For nearly a century, Scripps scientists have been studying the last frontier between society and nature, our coastal oceans. Today, nearly half the world's population lives within 100 miles of a coastline, and that population will only grow in the future. The impact of this growth is already having a significant effect on the health of coastal environments around the world. The issues of coastal ocean research include a diverse array of challenges. Questions about beach erosion, air and water pollution, marine ecology, and our fisheries. Here at Scripps, we're developing a new way of looking at our coastal oceans as a complete and integrated system. During the next half hour, you will learn about our present strengths and some of our future hopes for coastal ocean research at Scripps.
Among the original missions of Scripps Institution of Oceanography, when it was founded as California's first permanent marine laboratory in 1903, was to carry on a biological and hydrographic survey of the waters of the Pacific Ocean adjacent to the coast of Southern California. Over the ensuing years, coastal studies extended from San Diego's local beaches and the Scripps Pier and La Jolla shores to the rest of California's coastline, and later south into the Gulf of California and as far north as Alaska. Much of the early coastal research was descriptive. As investigations and collections continued, it became clear that understanding all aspects of the coastal environment would require applying science from many disciplines and making a commitment to long-term research programs. Temperature and salinity measurements have been made daily from the Scripps Pier since 1916, the longest-running record on the West Coast. In the late 1940s, the sardine fishery collapsed, and Scripps joined with the California Department of Fish and Game and the National Marine Fisheries Service in studies of the California ecosystem. Since then, the program has continued and now is considered the world's best and most comprehensive coastal environmental investigation. For nearly 100 years, Scripps scientists have studied the physics, biology, chemistry, and geology of coastlines with a focus towards issues of environmental quality, including questions about erosion, pollution, and ecology. Along the North American coastline, the most diverse and productive habitats are the kelp forests. Predominant is giant kelp, the largest and fastest growing algae in the oceans. Kelp forests provide refuge and feeding areas for many sea creatures, and in turn, attract extensive commercial and recreational fishing. Long-term studies at Scripps have shown that kelp forests are complex ecosystems, sensitive to both natural changes and human activities. Marine biologist Paul Dayton has helped lead this research.
The kelp forest was first studied in the very early 1950s when the state of California became concerned with the loss of kelp beds around Southern California. They contracted with Scripps to study that problem. And by the very early 50s, there was a large integrated program run out of Scripps at Point Loma. And some of those early divers, like Connie Limbo and Jim Stewart, recorded some of the most interesting natural history that we have for the kelp forest. They were in it very early, before it was disturbed, so they had a virgin community to study, and their papers are still very important. What has continued was the focus on the kelp plants themselves, the giant algae, the giant kelp. We started a continuous program with transects and the permanent sites in 1970, but they were placed on the same areas that Wheeler North and the early divers worked in 1954. So in a sense, we have a continuous research program that covers 50 years. I think that's the oldest diving research program in the United States.
Divers enter the water and swim down through the canopy of giant kelp to permanent study sites in the Point Loma kelp forest. They dive several days a week to count plants, measure growth, and look for signs of damage caused by storms or grazing animals. The divers measure the height and width of a giant kelp's holdfast, which is a mass of roots that anchors the plant to the seafloor. They record the data on plastic slates. The other parts of the giant kelp are the stipe, or stem, and the leaf-like blades that have air bladders that hold the plant upright. Each plant in the study is tagged and numbered so that the divers know which plant they are observing. A hole is punched in the base of the blade of a small kelp called laminaria. The distance that the hole moves from the stipe over the next month will indicate plant growth. A curious sheephead comes by for a closer look. Their mission accomplished, the divers return toward their skiff for the trip back to Scripps. Yet they know that as beautiful as the kelp forest is today, it is really only a ghost forest compared to just a few decades ago.
The difference is that in the early days, in the early 50s and 60s, the kelp forest was full of very large fish. They were all over the place. Now they're gone. They were full of abalones. The abalones covered the bottom. Now they're virtually gone. There were many large lobsters that are gone. Many small fish, cabezon and rock bass and kelp bass and sand bass are largely gone, and the ones that are here are much smaller. What's happened over time is that people have taken things from the kelp forest. We've all had this feeling that there's an animal. If I take this animal, there'll be another one there tomorrow or next week. Or perhaps if I don't take this animal, somebody else will. So we have slowly taken away all of the animals without understanding how difficult it is for these populations of animals to sustain themselves. And the problems aren't necessarily with human greed. The problems are, in many cases, simply lack of understanding, lack of proper management, lack of proper regulation. It's just bad luck. But now we have lost a lot of populations of animals, and it's going to be very difficult to get them back. I think that the main and the most important means of mitigating this damage is through setting up realistic, meaningful reserves. Reserves that are large enough so that the populations that we're interested in have a chance to recover and reproduce and send larvae out into the water so that other places can then be fished. These are sources of fish that can be taken elsewhere, but I think that it will be very difficult to recover these habitats without complete protection.
According to Dayton, marine reserves have proven successful where areas sufficiently large to allow for healthy reproduction and dispersal of larvae have been protected. And while fishermen may lose access to certain areas, they will reap the benefits outside the reserves as fish stocks thrive. Another area of growing concern along the coastline is water quality. California beaches and bays have the nation's most stringent public health standards and are monitored more than anywhere else. Unfortunately, they are also posted and closed more than anywhere else. Coastal contamination is primarily caused by two sources: urban runoff and sewage spills. A sewage spill can have devastating effects at the point of impact. However, in California, urban runoff is by far the greatest contributor to contamination. This runoff, called non-point source pollution, primarily is from storm drains. Scripps coastal oceanographer John Largier explains.
Back a couple of decades ago, we were worried about single industries, what we call point source pollution. And now it's really you and me and putting pesticide on our garden and fertilizer, and a little bit comes off everybody's yard and flows down to the ocean. And that's what we call non-point source pollution. That's the biggest problem. How do you manage that? And you might say, well, what's the effect on us? The effect is really twofold. One is at times it can threaten our health, the public health aspects. We might get sick if we go swim in polluted water. But probably the bigger and more challenging problem that we need to overcome is the threat to a sustainable ecosystem, to the animals and plants that live out here along the coast, and which of them are getting threatened, and can we keep a nice healthy environment going? What we've been doing recently in San Diego is deploying these thermistors or temperature recorders along the coast, right from the border up to La Jolla Shores. It's where our institution is.
Deployment of the instrument network begins in San Diego's Mission Bay. Staff Research Associate Melissa Carter heads out along the dock to one of the institution's coastal workboats, where technician Carrie Humphreys is ready to get underway. On board are the few simple supplies they will need for the morning's work. It's a cool, overcast day, typical of what the locals call the June gloom. They've set up a network of moored instruments at 12 sites along the San Diego coastline. About once a month, the team retrieves the data that have been collected at each site about the local fluctuations in underwater temperatures. Off Point Loma, Melissa and Carrie spot one of their buoys. They use a simple surface float anchored to the bottom by nylon rope about 30 feet below. Tied onto the rope are instruments that record water temperatures at the surface and on the bottom.
This is the instrument that we've been putting in the water. We can set it up to record temperature at whatever interval we want. It can record at 5 seconds or it can record at 10 minutes. We have them set for the work we're doing to read at 2 minutes, and the main reason we've chosen 2 minutes is because if we're looking at internal waves, those internal waves can last for maybe 10 minutes time. For recording for 2 minutes, we can get about 5 measurements that would let us know that an internal wave came through. 2 minutes also is long enough to give us 45 days worth of readings on these instruments, so we can only that gives us enough time where we can come back every month to service them or download the data. And to download the data, put it on the shuttle, and once we download the data, it shows a plot and it gives us an idea of what the temperatures look like over the time that the data's been in there. We can give it a rough check to make sure that our thermistor is working right and get an idea of the max and min over that time, and then later we'll plot it in a different program so we can overlay the different plots along the coast.
Back at Scripps, the researchers analyzed the data, looking for localized circulation patterns.
It looks like 5
Is
days.
the type of water motion we're seeing off Point Loma around the kelp forest different to what we're seeing at the beach at La Jolla Shores? And is it different to the beach down in Imperial Beach near the border? And the answer is yes. And that throws out the challenge to understand it better, to find out which locations along the coast are more susceptible to pollution problems than others. Well, ultimately, once you've described these patterns well, you can understand what's controlling the patterns, and you can predict some of the water motions and deal with the pollution problem much more effectively than we are.
In his studies so far, Largier has learned that while ocean circulation can wash pollutants away, it sometimes also traps pollutants in pockets along the shore. This accumulation can be particularly great in bays and harbors. The San Diego Bay is one of California's largest bays, and it's of tremendous economic importance to the city, state, and national government. Recreational boaters, cruise lines, shipyards, and the Navy all share its resources. On this day, we find another Scripps research team underway to sites where they are conducting biological experiments on the effects of pollutants on marine life at 6 locations in San Diego Bay. Dimitri Dehaene, a postdoctoral fellow from Belgium, leads the team. He is testing a research strategy that involves placing a common species of starfish from clean ocean areas into the bay, which contains pollutants from urban and industrial sources.
The San Diego Bay has been used by the US Navy as a big harbor and shipyard. As you can see here, we have big boats everywhere. San Diego Bay, therefore, is kind of polluted, and my research aims to see whether the back of the bay is more polluted than the outside of the bay. What I did for my research, I transplanted brittle stars, which is kind of a starfish, in cages, and I put them in different stations along the bay, and I will collect them every week during 2 months and see whether they accumulate pollutants, whether first they survive, and also see if they are a good indicator of the pollution in the San Diego Bay.
Dimitri and dive partner Phil Jondro suit up.
Ready? Okay.
All right. 1,
2, 3.
They swim along the boat line to the surface buoy with the Coronado Bay Bridge in the background. Underwater, they dive along the buoy's anchor chain to the bottom some 35 feet below. A plastic pipe grid holds the cages in place. A piece of mesh is removed. It has collected particles from the seawater directly above the cage that contains the brittle stars. The cages are cut free, kicking up a plume of the thick, loose sediments and placed into the dive bag. Part of the study is to measure the recently accumulated sediments, including levels of heavy metals and the presence of microorganisms. Another aspect is to withdraw pore water, the water that is saturated into the seafloor sediments, for chemical and biological analysis. Measurements are also made of the water chemistry, temperature, salinity, and turbidity from the bottom to the surface. This site survey completed for the day, the divers return to their boat. Later that night in the laboratory, Dimitri carefully opens the mesh cages to recover the brittle stars. The objective now is to evaluate each animal's exposure to pollutants by determining the types and quantities of materials in their tissues. Some brittle stars create luminescence, as this one does when chemically stimulated. The hypothesis is that the amount of bioluminescence reveals levels of toxicity. The next series of tests require the brittle star to be dissected. So Dimitri anesthetizes the animal and removes each arm so that he can analyze the effects of pollution on the light-producing cells. These arms are placed into a glass tube and put into an instrument called a luminometer that can accurately measure the light from the arms. A chemical is injected to stimulate light production. The numbers are the number of photons emitted. Other tests analyze the chemical content of tissue samples. Dehane discusses the results.
I put the brittle stars in different areas in the bay, and they didn't survive everywhere. In the back of the bay, the brittle stars did very well, and they didn't die, and they accumulated only a little of the pollutants. Whereas when you look at the sediment contains a lot of pollutants. But the pollutants are in the sediment and are not bioavailable to the brittle star. And that's something very clear when you go in the bay. The back of the bay is very turbid and you have a lot of suspension, a lot of particles in the water, which helps the pollutant to absorb to the particle and to sink into the sediment. So in the mouth of the bay, the water is clear and you don't have a lot of sediment. So the pollutants that are there don't have the opportunity to sink and to be attached to the sediment. So they will go into the organisms. And so the organisms will show a high level of pollutants in their tissue and they will be exposed to toxicity by that way. The mouth of the bay is more toxic than the back of the bay, but the amount of pollutants are different. You have more pollutants in the back, less in the mouth, but in terms of what is available, there is a huge difference. In the mouth of the bay, the pollutants, even if you have much lower level of pollutants, they are all bioavailable and they can be ingested by the organism, which makes a big problem.
DeHane's experiment points out the complexity of understanding the direct impacts of industrial pollutants on specific marine species and how localized water circulation and other factors can interact with these effects. Another aspect of coastal water quality under examination at Scripps is bacterial contamination. Most beach testing relies on finding fecal bacteria to determine if the water is unsafe. However, this standard does not detect other types of bacteria and viruses that may harm marine life or make humans ill. On this day, members of a group that studies marine microorganisms heads out onto the Scripps Pier. On the left, Richard Long, a postdoctoral researcher, and the right, visiting scholar Koji Hamasaki. It's a simple operation to collect 45 gallons of seawater with a boat hoist at the end of the pier. Trash cans may seem like odd buckets, but they're effective for the purpose, collecting a universe of microscopic marine life. In just 1 teaspoon are thousands of single-celled plankton, millions of bacteria, and tens of millions of viruses. Other water samples are collected in sterilized Niskin bottles when required. In the lab, the water is kept under artificial sunlight to keep the organisms alive. Small samples are drawn and taken to various workbenches. Some samples are labeled with antibodies that allow identification of specific bacteria under fluorescent light. This experiment involves inoculating seawater with small amounts of cholera bacteria to determine if cholera attaches to marine life and if it grows. The water is pumped out, with the filter trapping the microorganisms that can be examined with a fluorescence microscope. Director of the lab is Farouk Azam, a professor and marine microbiologist. They want to understand how microorganisms survive in the oceans and then apply that to disease-causing bacteria from contamination and pollution. Richard places a sample slide under the microscope and it is viewed on the computer. They are looking at specific disease-causing bacteria to discover where they find refuge and food sources in the marine environment.
There are microscale features such as dead and dying plants and animals, which may provide the environment for them to have explosive growths. And in order to understand their populations and persistent mechanisms, we need to understand this microscale structure of the ocean. And preliminary results show that actually these bacteria that are entering, they can do very well. Their ability to attach to micro environments, such as these dinoflagellates, we don't really know much about the detail of their lives in the ocean. So what we are doing is actually, with our experience in understanding how bacteria in the sea that are adapted to the ocean conditions, what kind of lives do they lead, what kind of adaptations do they have for survival and persistence and growth, to understand how bacteria entering into the ocean, such as through sewage, how they are likely to behave. And by using this context, we are learning, and we hope to learn more about how to think about contamination in a way that will allow us to make predictions rather than simple monitoring.
Preliminary work in Azam's lab shows that some bacteria survive much better in the oceans than predicted. And that raises concerns about current methods of water testing standards. California's coastal waters are invaluable assets. Foremost are the natural resources, an abundance of species and habitats, and the sand on our beaches. The ocean also serves as a source of food, recreation, and energy resources. And it supports industry, and provides a link for transportation throughout the West Coast, Pacific Rim, and beyond. The long-term health and sustainability of our coastal resources will require coordinated efforts to protect and enhance the entire ocean ecosystem. What's needed is a comprehensive approach to resource management in which citizens, governments, industry, and research all play a role. Understanding and monitoring California's coastal ocean remains a central mission of Scripps Institution of Oceanography, including investigations of water quality, pollution, and ecology. Research for the benefit of meeting today's needs, focused on assuring healthy resources for future generations.
Much of our research at Scripps is concerned with the sustainability of the coastal environment. In our studies, we will achieve common purpose with businesses, non-scientific organizations, government agencies, and individuals who are all interested in the well-being of our coastal oceans. By working together, we will connect scientific discovery with society's need to achieve a sustainable coastal environment.